This video is all about meiosis. So how many chromosomes do you have in a human sperm cell or a human egg cell? Well, a normal body cell has 46 chromosomes. So a sperm cell and an egg cell, because they're the ones that are going to be fusing together, as you actually see happening here, during fertilization, need to have half that number, or 23 chromosomes. So the question is, how do we do this? Well, meiosis is the process that's going to allow us to make these special sex cells or gametes that have half of the normal number of chromosomes. Oh, gametes, there it is. All right, so sperm cell, egg cells, these are produced by meiosis, which is a different process than mitosis, although as we'll see, it's pretty similar in a lot of ways. All right, so chromosome number is an important concept, and it refers to the number of chromosomes that each cell has. In ordinary body cells, we use a variable for chromosome number that is the letter N. And so in a body cell, that number would be 2N because you actually have 2 copies of every chromosome. So the number of chromosomes you have in like one set, that number is represented by the variable N, and this is different for different species. And of course, then most cells have 2 copies of every chromosomes, so they're going to be 2N. And the sex cells then are just a single N. And for humans, N equals 23. So cells that have 2 copies of every chromosome, we call those diploid, right? Di means 2, so we have 2 copies of every chromosome, or 2N number of chromosomes. And then the sex cells are going to be what we call haploid. Haploid means there's only one set of chromosomes, or 1N. So for example, a neuron, which is a cell you can find in your brain and in your nerves, versus a spermatozoa, the neuron is going to be 2N, and the sperm cell is going to be 1N. And in humans, those numbers are 46 and 23, right? Because N equals 23 in humans. There are 23 different chromosomes, or 23 pairs of chromosomes in a diploid body cell. All right, so mitosis and meiosis are a little bit different, like I said. And so this chart kind of quickly summarizes the differences between mitosis and meiosis. Mitosis is an asexual process that is going to result in letting us make exact copies of cells. So this is how your body grows. This is how your body repairs damage when you get an injury. This is how it replaces old cells that die off or need replaced for some other reason. And this is how you're going to make all those body cells, right? In mitosis, you just have one round of cell division. You go through prophase, metaphase, anaphase, telophase, right? PMAT. And then cytokinesis occurs, and you wind up with 2 cells. And those 2 cells are diploid, and there should be essentially the same as the cell that they divided from. Now, meiosis is a little bit different. For meiosis, this is a very specialized process that is only used for sex cells or gametes, right? Those sperm and egg cells. There are actually 2 rounds of cell division. So you go through PMAT and cytokinesis, and then you do another round of PMAT and cytokinesis, right? Prophase, metaphase, anaphase, telophase. And you should wind up with 4 daughter cells. But importantly, because we divide the cell twice without replicating the DNA in between, we actually wind up with those cells that are haploid. They only have 1 set of chromosomes, or 1N chromosome number at the end. All right. And this is kind of the big key difference in why sexual reproduction and meiosis are even a thing. And that's because while in mitosis, the daughter cells that result are identical, in meiosis, the daughter cells are different. They are unique from each other. And this is how sexual reproduction results in increased genetic diversity in a population. And that's really the entire benefit of sexual reproduction. So they're going to be different in their genetic material. They're not going to have identical DNA to one another. And that's going to be a benefit for whatever that population is, that population of organisms, that species that reproduces sexually as opposed to asexually. All right. So in the phases of meiosis, there are, like I said, 2 rounds of cell division. And in meiosis 1, there are some really important events called crossing over and the segregation of homologous chromosomes that help lead to that genetic diversity that we talked about. In meiosis 2, this is where we have separation of sister chromatids, and this results in haploid cells. And so this is very similar to what happens in mitosis, actually. Meiosis 2 is a lot more like mitosis, except that we've already gone through 1 round of cell division and shuffled up the genes a bit. So you can see here, these would be the cells that are going to be going through meiosis. So after DNA replications, we have our unreplicated chromosomes, and then these X shapes here are replicated chromosomes. Meiosis 1 separates and divides up those chromosomes, and so there's only 2 chromosomes here and 2 chromosomes here now instead of 4. And then in meiosis 2, we separate those sister chromatids. And at the end, our daughter cells, their nuclei, are going to each have 2 chromosomes, which is going to be half as many as the original one. You'll see that there's 1 big and 1 small in each one of these, right? 1 big chromosome, 1 small chromosome. Whereas in the original cell, there were 2 big chromosomes and 2 small chromosomes, right? This is our 2N cell, like our body cell. These are our 1N haploid gamete cells that result. All right, we're going to go through the different phases again here. If you want to pause and look at this diagram, you can. I'm not going to go through it because I'm going to go through each individual phase in a little bit, but this is kind of an overview of meiosis 1, prophase, metaphase, anaphase, telophase, and cytokinesis. And then this is kind of an overview of meiosis 2, prophase, metaphase, anaphase, and telophase, and cytokinesis, right? And you'll see that at the end we have N haploid cells right here. All right, moving on, let's go ahead and look at the individual phases. So in prophase 1, our nuclear envelope begins to break down and the chromosomes condense. Centrioles start moving to opposite poles of the cell and a spindle begins to form. So you can see that happening right here. We have these centrioles going to opposite sides of the cell. We have our spindle fibers forming. Our nuclear membrane is breaking down. And the most important thing that happens here is that homologous chromosomes pair up. So these homologous chromosomes pair up and they do something called crossing over. And crossing over is where the chromosomes actually swap bits of their DNA. And this is really important for increasing genetic diversity in sexually reproducing organisms. Then we have metaphase 1, and this is where our spindle fibers attach to the paired homologous chromosomes. The paired chromosomes line up in the equator, and then they will be able to separate in the next one, right? So here are those paired up homologous chromosomes that have done crossing over. They've swapped some of the genetic material, right? Only with homologous chromosomes, and they are now going to separate in the next phase. Now, really importantly, one of those really important genetic principles that was actually discovered back in the day hundreds of years ago by Gregor Mendel, the principle of independent assortment, that is due to the fact that these genes that are on different chromosomes can line up in different ways. So you'll see that, for example, on this pair of chromosomes, the red one, what we call the maternal homologue, the chromosome from mom, is on this side of the cell, and the blue one's over here. But on this one, it's the opposite, right? The blue one, the paternal homologue, the one from dad, is on this side of the cell, and the red one is over here. So these can line up any of those different ways. It just depends on how they wind up arriving in metaphase 1. And so they can be in all sorts of different combinations. And because of that, genes that are on different chromosomes are inherited independently. They're independently assorted. And this is what allows us to make Punnett squares, essentially. If it weren't for the principle of independent assortment, you wouldn't be able to do 2-factor, 3-factor plus crosses. You'd have a lot more genes that are all inherited together. And that's just not what happens in reality because of that principle. All right, then we have anaphase 1. So this is where our chromosomes of each homologous pair start to separate from each other, then move toward opposite poles of the cell. So you see that happening, right? They are now moving toward those centriole structures at the poles of the cell. And then telophase 1 in cytokinesis, where the spindle fiber breaks down and a new nuclear membrane forms, and the cytoplasm of the cell divides and 2 daughter cells result. So we have that happening here, right? We can see that the cytoplasm starting to divide right here in what we call a cleavage furrow. We can see that the nuclear membrane starts reforming, and yeah, and so we're gonna, almost like as if we're gonna form 2 new cells and 2 new nuclei. But that process that begins right here in telophase 1 cytokinesis immediately gets reversed in our next phase, which is the first phase of meiosis 2. So we have prophase 2, where that nuclear envelope we just started building starts breaking back down again. And that might seem kind of silly. Why would it do that? Why would it reform a nuclear envelope just to break it down immediately? Well, because meiosis evolved from mitosis, and that's what happens in telophase of mitosis. So it might not make much sense, but it works. And as far as evolution is concerned, as long as it works, it's fine. So there you go. So this is going to look, meiosis 2 is going to look a lot like mitosis, except we're starting with a smaller number of chromosomes, right? We're going to start with only 46 chromatids in humans, for example, as opposed to 92 chromatids, which are those copies of the chromosomes. So very similar to what we see in mitosis, right? Their nuclear envelope breaks down, spindle fibers begin to form, centrioles move to the poles of the cell. Just like in in prophase of mitosis, metaphase 2, spindle fibers line up the sister chromatids of each chromosome along the equator. So just like this, just like we've seen before, right? Anaphase 2, they separate just like in mitosis. So they're traveling to opposite ends of the cell. You'll notice though, these diagrams, right? There's 2 cells for each one of these. Don't forget, there's 2 cells going through meiosis too. And then at the end, we have telophase 2 and cytokinesis, where our spindle breaks down, the nuclear membranes start reforming. We have the cytoplasm start to divide, right, in each one of these pairs. And so we're going to wind up with 4 cells at the end of this. And you will note that if you look at the mix of red and blue DNA here, which is representing DNA from mom and DNA from dad, that all of these cells have slightly different combinations of red and blue DNA because of the fact that each of them is only inheriting half. And we've shuffled those up through those processes in prophase 1, where we did crossing over, where the chromosomes swapped bits, and in metaphase 1, where they randomly aligned, right? Just the random alignment. And those 2 things together lead to the independent assortment of genes, right? And so then, therefore, the independent inheritance of genes, meaning that just because you get an eye color gene from mom doesn't mean that actually was grandma's or whatever. Doesn't mean you're going to get grandma's toe shape gene, right? Or grandma's hair color gene, or grandma's irritable bowel syndrome gene. Okay, you might get grandpa's toe shape gene or grandpa's hair color gene, right? It just depends on how those chromosomes lined up. They're going to be inherited independently. They're not all going to be inherited as a whole set. So in the parents, the genes get shuffled up before they get passed on to the offspring. And that's really important for increasing genetic diversity. It's really important to allow for siblings to be different from each other, right? So you are different than your siblings if you have siblings, partially because the genes in your parents got shuffled before they were passed on to you. And it's because of these important processes in meiosis. All right, so homologous chromosomes. I kind of talked about this a little bit already. The really important key feature here. These are chromosomes that are similar to each other, but they're not identical, right? So every chromosome every chromosome pair that you have, right? Those 23 chromosome pairs, there's the paternal homolog, meaning the one you got from dad, and there's the maternal one. And you'll see that along these, you have genes that are the same gene in the same location, right? The same locus on the chromosome, what it's called, the same locus has the same information encoded in it, except you can have different versions of them. So for example, this one, the G gene, whatever that is, maybe it's a growth gene that kind of determines how big you are, how tall you are, how thick your bones are, something like that, right? You can get a dominant version from one parent or a recessive version from another parent, right? Maybe this one makes you taller, okay? This one makes you shorter, for example, right? But you could also have the same copy, right? Maybe this is that FH1 gene you looked at earlier when we did Punnett squares. Maybe this person's going to be homozygous for FH1, so they're going to have really high cholesterol. That's possible, right? Because they have 2 copies, one from each parent. All right, then we have fertilization. Fertilization just occurs when a sperm cell and an egg cell fuse, right? And that fertilized egg cell is called a zygote. So we have our N equals 23 in humans, sperm cell, our N equals 23 in humans, egg cell, and together they're going to give us a zygote, and that's going to have that 2N chromosome number, which in humans, of course, is 46. All right, we'll come back to non-disjunction in the next video.